ترتاح وgood luck.
شكرا. can I share my screen هون؟ بتشوفوا الباوربوينت هلا؟
تمام هيك ممتاز.
ايه شكرا. So اليوم بدنا نحكي على this big title وما بعرف كيف أقول هدول الكلمات بعربي so بدي أحكي بإنجليزي لهذا التوك. بس أول بدي بلش مع هذا اللاب. أنا كنت أشتغل in the Castro and Lowenstein lab last year. هلا ما عندي كتير وقت يمكن بدي بلش بعد 6 شهور. But in their lab they study the epigenetics and immunotherapies for different types of brain tumors. And today we're going to talk about their paper about immunotherapies in gliomas, which is a specific type of brain tumor. And the first thing I wanted to go through is the introduction. What they focus on is this enzyme called isocitrate dehydrogenase, IDH, and they have two main isozymes. One is IDH1 and one is IDH2. They do the same thing, they just do it in different locations. So IDH is responsible for converting isocitrate into alpha-ketoglutarate. And IDH1 works in the cytoplasm, and IDH2 does this in the mitochondria. Mutations in this enzyme are really common in gliomas. And in IDH1, it's usually an arginine to histidine mutation at the 123rd spot. And then in IDH2, it's usually a mutation at the 172 spot, but it's less common than in IDH1. And what this mutation does is actually leads to a gain of function in alpha-ketoglutarate, and it converts it into something called 2-hydroxyglutarate, this 2-HG. And there's been a lot of studies that show that 2-HG actually impacts the tumor microenvironment of mutant IDH1 gliomas, and it leads to things like decreased programmed death ligand-1 expression, decreased inflammation, and decreased immune cells. So the body's less able to fight the cancer because there's not as much of an immune response because of this 2-HG. And usually myeloid cells represent the major immune cells in the glioma microenvironment. So a big part of this paper is going to figure out how 2-HG modulates the immune microenvironment of a glioma and what we could do in order to counteract that immunosuppression and provide better immunotherapies. So this is kind of what I just talked about, the goals of the paper, where the tumor microenvironment of mutant IDH1 is very different than the tumor microenvironment of wild type IDH1 without the mutation in the enzyme IDH1. And what they found is that one of the biggest types of cells that suppress the immune system are these things called granulocytic myeloid-derived suppressor cells, so MDSCs. And I'll just refer to these as immunosuppressive myeloid cells. And part of this paper, they're also going to explore the therapeutic response of mutant IDH1 gliomas to this new immune therapy called TK-FLT3. It has a large name, but it basically attacks one of the enzymes that's important for cell proliferation, tyrosine kinase. And then from here, I just wanted to start by going through each of the figures because they kind of take us through the whole paper. And starting with figure 1, so first I wanted to uncover the impact of mutant IDH1 in the context of glioma with an ARTX and TP53 deficiency, which is two very common inactivations found in glioma, so they wanted to make it more realistic. And they wanted to see the impact on mutant IDH1 on these immune suppressive myeloid cells and how it would respond to immunotherapy. So what they did, as you could tell in figure B, is that they had two separate sets of mice, ones with wild-type IDH1 gliomas and others with mutant IDH1 gliomas, and they treated them with the drug of choice, this TK-FLT3 immunotherapy, or they gave them nothing. And then they gave them this Ly6G depletion, which depleted the myeloid cells to see if that would confer a survival benefit, because the myeloid cells they thought were inhibiting some of the immune cells. And what they found was that treatment with this TK-FLT3 prolonged survival of both mutant IDH1 and wild-type IDH1 tumors. But the change in mutant IDH1 was even stronger compared to just wild-type IDH1. And also combining the TK-FLT3 with Ly6, which depleted the myeloid cells, it helped the wild-type survival increase, but not the mutant IDH1 survival increase. And then they wanted to repeat this study with a marker that would inform them on the specific response of T cells. So they used this thing called OVA, which you see in figure E at the bottom right, and it allowed them to quantify the tumor-specific CD8+ T cells. And they found that the gene therapy that they used increased the number of tumor-specific CD8+ T cells infiltrating the tumor microenvironment in both wild-type gliomas, IDH1 gliomas, and mutant IDH1 gliomas. So this confirmed going back to the purpose of this, this confirmed that the TK-FLT3 therapy was working, and depleting the myeloid cells helped with in wild-type gliomas, but not the mutant gliomas. And you can see the difference in survivability in figure C and D, where you have the wild-type tumor on the left, and you see a significant difference between the red and the blue line, where the red line is the gene therapy with the myeloid depletion, and the blue line is without the myeloid depletion. And then in the mutant cells, you see no difference between myeloid depletion and no myeloid depletion, but you see that the gene therapy really helped tumor survival by a lot. And then the next thing they wanted to do in the study was actually study the phenotype of the myeloid cells that were being impacted by mutant IDH1. And there are a lot of cell markers that they used to quantify the types of cells that they were finding. And what they found was that there was an expansion of these immunosuppressive myeloid cells in the tumor microenvironment of the mutant IDH1 gliomas compared to the wild-type IDH1 gliomas. And this is just some flow cytometry studies showing that as well. And then they did further analysis of those myeloid cell populations, and they found that in the mutant IDH1 subset, that most of these myeloid cells belonged to the granulocytic population, which you could see in figure C, where Ly6G is indicative of granulocytes. And you can see that compared to wild type, there's a lot more granulocytes in the mutant than wild type. And then they wanted to test if this increase in myeloid cells was associated with the growth of the tumor. So they studied the frequency of the myeloid population at the mid-stage of tumor growth, which I think, yeah, I put it here as 12 days after they implanted it into the brains of these mice. And then they also recorded the frequency of the myeloid cells when the mice started presenting with symptoms. And as you can see in figures D and E, increased frequency of these myeloid cells was associated with increased tumor growth, especially in the symptomatic stage in the mutant IDH1 gliomas and not in the wild type IDH1 gliomas. And also they did a whole body analysis of these mice, and they found that the expansion of the granulate cells, of the myeloid cells, was not only in the brain tumors, but it was also around the whole body. So they looked at the bone marrow, they looked at the spleen, and they looked at just the serum, so the overall blood circulation, and they found an elevated myeloid population in the mutant IDH1 mice and not in the wild type IDH1 mice. And they then wanted to test whether these granulocytes, so a subpopulation of the myeloid cells, there are a lot of confusing terminologies here. They wanted to see if they were immunosuppressive against T cells. So they used this thing called SIIN, which essentially just leads to the proliferation of T cells. And you could see that in the bottom left of the graph, where you see they only have T cells, no proliferation, and then they add this proliferative agent, and it leads to a lot of T cell growth. And then when they added these myeloid cells from the tumor microenvironment of the wild type and mutant IDH1 gliomas, they saw that in the wild type, they were immunosuppressive. So they were suppressing T cell function and immune function in the wild type IDH1 gliomas. But with mutant IDH1 gliomas, they were not suppressing T cell proliferation at all. As you could tell here, they remained at close to 100%. So what this suggested was that the expanded granulocytes in mutant IDH1 tumor environments were not immunosuppressive, which is important when you think about immunotherapies and the difference in how you could treat mutant IDH1 gliomas versus wild type IDH1 gliomas. This suggests that we could harness whatever non-immunosuppressive properties of mutant IDH1 uses and reverse the phenotype of wild type IDH1 and hopefully use immunotherapies with that type of a glioma. And this is kind of what I talked about, where they found that in mutant IDH1 tumors, they induce some sort of different altercation in hematopoiesis in the bone marrow to make these different types of granulocytes. And this is a diagram depicting that. They did an analysis of the, I'm getting ahead of myself. They analyzed the myeloid cells that they isolated from the mutant IDH1 tumors, and they're, like our previous experiment suggested, they have less immunosuppressive properties than the wild type IDH1 tumors, and this transcriptome analysis indicates that as well. They found decreased expression of all immunosuppressive genes associated with the immunosuppressive myeloid cells in the mutant IDH1 tumors compared to the wild type IDH1 tumors, which is just adding, it's making their claim that these types of myeloid cells are immunosuppressive. It's making that claim a little more robust. And then they wanted to analyze these cells on a single cell basis. So they did single cell RNA sequencing. And what they found on the left side in figure B, this is the tumor microenvironment of mutant IDH1, they found 14 distinct cell clusters with that were indicative of known immune cell types. And the biggest out of all of these, you see some macrophages, monocytes. The largest population was comprised of granulocytes. And within the granulocytes, they found 3 distinct clusters, and they just called them C1, C2, and C3. And I don't show the analysis for wild type IDH1, but wild type IDH1, they also had a large population of granulocytes, but they only had 1 cluster, which was C1. And they did further experiments on each of these clusters, and they found that C1 was the cluster that was inhibiting T cell proliferation, suggesting that it was those immunosuppressive myeloid cells of the tumor microenvironment. So this experiment in figure F is really similar to the previous one where we showed T cell proliferation, and they used the same T cell proliferative agent, CIIN, and they show that compared to C2 and C3, C1 was the only cluster that reduced T cell proliferation, further indicating that this is the immunosuppressive cluster. And then they wanted to look into the specific marker that could differentiate the immunosuppressive granulocytes from the non-immunosuppressive granulocytes. So they did further studying of the different markers on the surface of these inhibitory cells, and they found that CD16 and CD32 positive myeloid cells were the they were markers of the cells that were inhibiting T cell proliferation compared to CD16-32 negative cells. These were non-immunosuppressive. So they succeeded in their goal of finding a differentiating factor between these two subsets of cells. And again, they use SIN in figure E here, where they showed that the 1632 positive cells were those that were suppressing the proliferation of T cells compared to the CD1632 negative ones. These were not immunosuppressive, just allowing them to categorize this subtype further. And there's not a lot to figure 6, but I thought it was important to bring up that they just had to everything that was just discussed were in mouse models. And at Michigan, we have access to a lot of patient samples, and they wanted to reconfirm all their findings in patients because that's what matters. And first, they started by showing that this immune suppressive myeloid cell type, it was more prevalent in the wild type IDH1 than in the mutant IDH1 gliomas, which we suspected which we found in mice, but they just confirmed it here in humans as well. And that's what they're showing in this bar graph in figure E, where the proportion of immunosuppressive myeloid cells is much higher in the wild type gliomas compared to the mutant IDH1 gliomas, and it's statistically significant. And now they've established that the granulocytes in the majority of the granulocytes in mutant IDH1 gliomas are non-inhibitory. And they wanted to test the theory that this change in phenotype and function is due to changes occurring at the epigenetic level. So there's some epigenetic programming going on in the mutant IDH1 tumors that prevents them from being immunosuppressive. And what they did, they looked for enrichments of H3K4 ME3, which is associated with a transcriptional activation. And they found that this gene, colony stimulating factor 3, was the one epigenetic, it was the one gene that showed a significant enrichment compared to wild type IDH1. And G-CSF, colony stimulating factor 3, is responsible for the production of G-CSF, which is granulocyte colony stimulating factor, which promotes the proliferation of granulocytes. And here in figure 7, they wanted to ensure that the epigenetic enrichment of CSF3 was associated with an increase in the actual protein that it produced, which is this granulocyte stimulating factor. And here is a confirmation of those findings, where you see the enzyme of interest, G-CSF, on the y-axis, and you have wild type versus mutant IDH1 gliomas. And they found a significantly elevated level in mouse serum, mouse conditioned media, and human conditioned media, which is showing that this increased enrichment at the epigenetic level is actually leading to an increase in this colony stimulating factor protein. And then this is data from actual human glioma samples, which is showing the same thing to a much lesser significance because of how ideal the previous experiments were. This is in an actual glioma, and it's still showing that there is greater CSF3 expression in mutant IDH1 gliomas compared to wild type IDH1 gliomas, which is indicative of this epigenetic programming occurring in mutant IDH1. And they wanted to uncover the source of this granulocyte colony stimulating factor within the mutant IDH1 tumors. And they did an analysis of various different cell types within the mutant IDH1 gliomas, and they found that the stem-like cells had the highest expression of CCF3, which was indicative of this cell type being the most important for the unique properties of mutant IDH1 gliomas. And since they were stem cell-like, it was important to point out that the undifferentiated glioma cells in the mutant IDH1 tumors were the cells that were most responsible for the phenotypic remodeling of the granulocytic myeloid cells. So this immunosuppressive, or this non-immunosuppressive property, came from these undifferentiated cells. And G-CSF is also important, not only because it produces these granulocytes that are non-immunosuppressive, but it also promotes the survival and differentiation of myeloid progenitor cells into neutrophils. So they found that G-CSF is important for creating this non-immunosuppressive phenotype. So what the lab was thinking is that if you suppress the production of that enzyme G-CSF, you should also reverse the non-immunosuppressive phenotype of mutant IDH1 tumors. So this alpha G-CSF is essentially like an antibody that inactivates the colony stimulating factor and would lead to a depletion of the granulocytes in mutant IDH1. So what they did was they studied T cell proliferation in normal mutant IDH1 gliomas, which we know doesn't really inhibit T cells, which we see in the red bar graph. But then when they inhibited the granulocyte colony stimulating factor, it did deplete the T cells. So it acted more like wild type IDH1 gliomas. And this showed that likely where the mutant IDH1 glioma is getting its non-immunosuppressive properties is from this granulocyte colony stimulating factor. So this gave them a tangible target if they wanted to take these findings and apply them to wild type IDH1. This was a tangible enzyme that they could target, inhibit, and hopefully reverse the immunosuppressive phenotype of wild type IDH1 tumors, allowing them to be more susceptible to immunotherapies. And something else that the authors pointed out, which was interesting, all these graphs on the right show different types of glioma tumors. And the lower grade glioma, which is what they're studying in this paper, is the only tumor where patients have a better survival rate if they express CSF3 compared to patients with low CSF3 expression. Every other tumor is different, and they did not investigate the reasoning behind this, but it might largely be due to this immunosuppressive phenotype that it inhibits. And lastly, they tested the findings that I just alluded to, where they wanted to see if suppressing G-CSF alone is sufficient to induce the expansion of these non-immunosuppressive neutrophils, which would be important if they wanted to apply it to the wild type. If you suppress the G-CSF, then it becomes non-immunosuppressive and you could use immunotherapies. And in 10E, you could tell that this is all in wild type gliomas, and when they treated these myeloid cell populations with granulocyte colony stimulating factor, or they blocked it with this antibody, you could see that it lost its immunosuppressive phenotype. They weren't able to deplete the proliferating T cells. So now, just by inhibiting G-CSF, they harnessed that non-immunosuppressive capability of mutant IDH1 gliomas and put them and enhanced it in the wild type IDH1 gliomas. And then finally, they went back to that first experiment they did where they tested the efficacy of this gene therapy, TK-FLT3, on prolonging patients or prolonging mouse survival in this case. And this time, they combined it with the granulocyte colony stimulating factor inhibitor in wild type IDH glioma. And compared to the blue line, which is just the gene therapy, which we know is sufficient enough for increasing patient survival, adding this granulocyte colony stimulating factor inhibitor led to a significant increase percent survival in the mice with wild type IDH1 gliomas, which is really promising. That was the whole purpose of this study, essentially. And I thought that this summary at the end of their paper was a very succinct way to put everything, because we went through 10 long figures with a lot of findings. So through epigenetic reprogramming caused by 2HG, which we know comes from mutant IDH1 gliomas, not wild type, they found that those undifferentiated stem cells in the mutant IDH1 cells expressed this high level of CSF3 that came from epigenetic programming, and that produced granulocyte colony stimulating factor, which shifted hematopoiesis in the bone marrow and spleen, and it made more of these granulocytic myeloid cells, which are non-immunosuppressive, which allowed for enhancing the efficacy of immunotherapy. So taking the essence of this sentence and finding that targetable thing, which is G-CSF, they're able to take this finding and lead to hopefully promising results in wild-type IDH1 gliomas. And this is more of a drawn-out wrap-up to the paper. So first, they found that the myeloid cells in mutant IDH1 gliomas are not immunosuppressive in the mutants compared to the wild type. And they found that depleting the myeloid cells further enhances the efficacy of immunotherapy within the wild type IDH1 glioma, which is what we showed in figure 10. By inhibiting G-CSF, they did not inhibit T cell proliferation. And then within the granulocytes in IDH1 gliomas, they found 3 distinct cell clusters, C1, C2, C3, and they found that C1 was the immunosuppressive subtype, and that wild-type IDH1 gliomas only had this immunosuppressive subtype of granulocytes. And then they found that the reason that G-CSF was upregulated in mutant IDH1 gliomas was due to epigenetic reprogramming, and the cells that they found that were producing the most CSF3 were expressed SOX2 and SOX4, which are markers of stem-like cells. And then lastly, this conclusion statement is similar to the previous slide, where they found that granulocyte colony stimulating factor reprograms hematopoiesis in gliomas, and it promotes the production of these non-immunosuppressive myeloid cells, which can be harnessed to enhance immunotherapeutic efficacy. So the next steps, this paper was released in 2021, I believe, and a lot of these human trials take a long time, and we have some other human trials that they're currently working on. Their main focus right now is proving the efficacy of this gene therapy alone, TKFLT3, for the treatment of pediatric gliomas, I believe. So hopefully we see the fruits of this happen within the next decade, but from talking with my principal investigators at Michigan, I know that these processes are very slow. But any questions?
شكرا كثير ميشيل على شرح البيبر. هي يمكن شوي صعبة كانت أو بحال حدا عنده أي سؤال فينا هلا يسأل، بحب كمان بحال يسأل بالعربي ويكون معنا أكيد الموضوع. تمام. هلا بحال ما حدا عنده حبلش أنا بسؤال. بصراحة شوي الميثودولوجي ما كانت واضحة بالنسبة إلي. Did they mention يعني how did they apply the therapy to the mice?
Yes.
Was it local يعني ولا was it ما بعرف intravenous or شيء؟
I think they did. I think they injected the therapy directly into the glioma when they did it in the mice. But they had different conditions in which they did this. They had conditioned media, so just like petri dishes, and then when they did it in the mice with gliomas, they injected it into the glioma itself.
Maria عم تسأل, can you tell us if there was any limitation while doing this study?
From my time in the lab, one of the biggest limitations is actually getting the mice with the proper phenotype and the genotype, because you can't purchase them. You have to breed them yourselves, and everything has to be perfect. And a lot of them don't survive because you're implanting brain tumors in them. So I think the N, probably the sample size, is the biggest limitation.
تمام. الصراحة أنا كمان عندي كام سؤال، ما بعرف إذا فهمت الموضوع مظبوط، بس was يعني the therapy only effective in mutant IDH1 glioma?
Yeah, so they know that this therapy works in mutant IDH1 gliomas, but they didn't know why it didn't work in wild type. And then through this analysis, they found that it worked in mutant IDH1 gliomas because the mutant gliomas were not inhibiting the immune system of the patient that they were in. And the purpose of this paper was finding out why their immune system was not inhibited. And once they found that granulocyte colony stimulating factor, the G-CSF, and they turned it off in the wild type, that allowed the patients of the wild type glioma to have a normal acting immune system and respond to the immunotherapy. So at first it was only the mutant IDH1 gliomas, and then this paper was understanding why and then applying it to wild type IDH1 patients.
And the G-CSF was only also present in mutant IDH1 gliomas?
Let me double check. It's present in both, but it was upregulated or it was downregulated in mutant IDH1 gliomas. Yeah.
So I'm just asking because so when you showed that graph with different tumor types, we found that glioblastoma that it did not really make a big deal when the G-CSF was found in glioblastoma or not. Maybe because 90% of glioblastoma are IDH are wild type IDH1. I'm not sure. What do you think?
That's a good point. That's a good question. From my understanding of glioblastoma is that it's a whole array of different cells, and that adds to the complexity of treating it, where these gliomas are a little more contained. But I wonder, so do you mind repeating that one more time?
So I'm just saying that glioblastoma, 90% of the tumors are wild type. So maybe they cannot be, and it's always a tricky thing that, so I personally faced this in the lab when we established tumor microenvironment in the organoids, for example. Yeah, the response to the immunotherapy went below normal standards. So it was really interesting when you started that we can make a gene therapy and maybe regulate the tumor microenvironment this way. But then when it does not work in wild type, this was my question. So if it does not really work in wild type, what should we do then? And is it because of the G-CSF?
Yeah, that's a really good point. I don't know how translatable this is from glioma to glioblastoma, but in glioma, at least in the lower grade glioma, it's a lot more simple because they found this one G-CSF, the colony stimulating factor that was responsible for making non-immunosuppressive phenotype. And once they inhibited it, it worked, and the immune suppression disappeared. I'm not sure if this has been tried in glioblastoma, but I would guess that it's more complicated with glioblastoma, that there's not just one protein that's really responsible for having a poor immune response to immunotherapies. But ideally, they would repeat the study in glioblastoma and find whatever marker or whatever protein is causing it to be invisible to the immune system. I don't know, with your work in glioblastoma, is it immunotherapies that you primarily do, or is it something else?
So basically we try CAR T cells, so yeah, kind of an immunotherapy. And yeah, it's always tricky to balance between the tumor microenvironment and the immunotherapy. It's a very smart tumor to treat. Too smart. Okay, so we have another question from Arla. She says, could you explain the prognosis part briefly again?
Which prognosis part? About the improved survival?
Yeah.
Oh, okay. So there were two of those. I'll share my screen again. So there were two graphs where we showed increased survival. And in this first one, this is just in the mutant IDH1 group and the wild type IDH1 group. This is not with any G-CSF manipulation. It's just the subtypes. And they just wanted to test the efficacy of their gene therapy just to show that it worked to begin with. And they also wanted to deplete all the myeloid cells, not just the immune suppressive ones, because they didn't know at this time which were immune suppressive. And they showed that in the wild type tumor, this gene therapy, the TKFLT3, led to increased survival overall compared to the mice that didn't get the gene therapy. And then adding that myeloid depletion in the wild type only led to an even further increase in survival, likely because they were inhibiting the immunosuppressive qualities of the wild type tumor. And then very similar things happened in the mutant IDH1 tumor, but because it already because it already can't suppress the immune system, adding the myeloid depletion didn't make a difference between the two drug groups, because it wasn't conferring any benefit. And then the last time I showed a improved survival chart was in wild type IDH1 tumors. So the wild type IDH1 tumors suppress the immune system. First, on the left, what they wanted to show was that they could harness the ability of mutant IDH1 tumors to allow an immune response. They wanted to show that they could do that in wild type IDH1 by adding the inhibitor of G-CSF. And they showed that in figure E at the bottom left. The two blue graphs, the two blue bars, are not with inhibition of granulocyte of G-CSF, and you see that the T cells aren't able to proliferate. But then after they inhibited it, the T cells were allowed to proliferate. There was a proper immune response in wild type, showing that the immune system might be better able to respond to immunotherapies. And they showed that then by improved survival in the mice. I can't see the message. Is that a better explanation?
I think so. إذا على لسه في شيء ما واضح فيك أكيد تخبرينا. I think كان كل شيء تمام. ماريا عم تسأل إنه she was wondering why they couldn't know the reason of patients with high G-CSF, their prognosis were better than patients with low CSF.
Yeah, I think that goes back to Mato's point about glioblastoma and why it might not be why the immunotherapies don't work on it. And I think it's because in low-grade gliomas, uniquely, high CSF3 leads to non-immunosuppressive qualities. But it seems like that might be very unique to low-grade gliomas, where in these other types of cancers, high CSF3 is not the reason why immunotherapies don't work. There's some other thing that is not clear. Have they applied? So the last question, they have not applied it on humans yet. They're just starting with that initial gene therapy, the FLTK3 thing, because they want to show that that works first. And they're starting with low-grade pediatric gliomas. And I think slowly over the next 10 years, they're going to add this granulocyte inhibitor and see if that helps with immunotherapy.
اوكي شكرا كثير ميشيل. بعد حدا لسه عنده اي اسئله او اي كومنتس للدسكشن؟ اكيد لسه معنا تقريبا 5 دقائق او شيء.
شكرا. Thank you for all the questions. بعرف هاي كان كثير صعبة بس I thought it was interesting.
اوكي اذا ما حدا عنده ولا سؤال، حابه بس استغل اخر كم دقيقه معك ميشيل واسالك what are your future plans وشو الفيلدز اللي حابب تكفي فيهم؟
هلا أنا تاني سنة بميديكال سكول بميشيغان. هاي السنة نحن بالمستشفى وعم نشوف كثير شغلات. So أنا عملت OBGYN Pediatrics وسيرجري. خلصت family medicine. حبيت surgery كتير. ما بعرف إذا I have to do internal medicine. If I like the body, then maybe I'll do the body. بس هلا I think I like pediatrics and neurosurgery, so that's what I'm doing now. So hopefully in 2 years I'll match into neurosurgery if I still think that.
Okay, good luck.
شكرا.
وشكرا كثير لك، كانت محاضرة عن جدا ممتعة. شكرا كثير لكل الناس اللي حضروا، والمحاضرة أكيد مسجلة للناس اللي ما كان عندهم وقت وما سمحت يعني ظروفهم أن يحضروا هلا. لينك رح نبعته كالعادة على الجروب، وإذا حدا عنده أي شيء حابب يضيفه أكيد يعني هلا الوقت مناسب. اوكي تمام، معناتها شكرا كثير ميشيل، ونتمنى لكم نهار سعيد للكل وويكند سعيدة، ونشوفكم ان شاء الله الشهر الجاي.